Bismillahirrahmanirrahim. Good morning from Lahore, Pakistan. I am Dr. Javed Iqbal Khokar, Professor of Forensic Medicine and Toxicology, and I am starting the new topic asphyxia, and this is the first lecture on this topic. The learning objective of this lecture will be that I will be discussing what is asphyxia and what is the difference between hypoxia, anoxia, and asphyxia, and what are various types of hypoxia. The physiology, the biochemistry, and pathology of asphyxia will be discussed. And I will also be discussing the signs of asphyxia, which are specific signs depending upon the type of interference, and the non-specific signs which appear in almost all asphyxial deaths. It has been seen that the asphyxial deaths, 5% of all unnatural deaths are predominantly by the violent asphyxial deaths. And when we talk about the asphyxia, it's a Greek word which means pulseless. A means negative or absence, and asphyxia means the pulse. And it results from the lack of oxygen, either partial, which means hypoxia, or complete, that is anoxia. And by definition, when we say asphyxia, asphyxia is a condition in which the supply of oxygen to the blood and to the tissues has been reduced below the normal physiological level by any interference to the process of respiration. And the term asphyxia in forensic medicine is restricted to those forms of oxygen lack which results from mechanical interference to the process of respiration. The supply of oxygen and its utilization is the basic requirement of each cell for life. And such anoxia can occur at any level, starting from the deficient oxygen in the atmosphere to the impaired utilization by the tissue cells. Anoxia, asphyxia, and hypoxia. What is the difference between these terms? Anoxia is complete absence of oxygen supply to the body tissues, whereas hypoxia means reduced quantity of oxygen which is available to the body tissues, and asphyxia means the interference with the process of respiration. Now, what are the causes which leads to interference? They are predominantly, number one, either there is no oxygen in the environment, or the environment has been replaced by irrespirable gases. Gases are there, but they are not for the respiration. Then either it is blocked airways at any level, the airways are blocked, or the diseased lung, the oxygen transfer mechanism is interfered because of disease of the lung. Then defective pulmonary or systemic circulation, that is the carrying capacity of the blood is reduced by the circulating volume which is interfered with, or defective red cells transport, that means the oxygen carrying capacity of the red blood cells is interfered with. And then finally the tissue poisoning, because the oxygen has to be utilized in the metabolic processes at the tissue level, and then the tissue poisoning is the ultimate asphyxia cause. Then what are various types of asphyxias? Either it is hypoxic hypoxia, or anemic hypoxia, stagnant hypoxia, or histotoxic hypoxia. Hypoxic hypoxia means that there is a reduced transfer of oxygen at the level of lungs, which means the reduced transfer from the atmosphere to the lungs, and hence from lungs to the blood. Then anemic hypoxia is the condition in which the blood is incapable of carrying oxygen to the tissues. Either the volume of blood has been depleted because of hemorrhage, or the oxygen carrying capacity has been reduced due to various other reasons like carbon monoxide poisoning, and stagnant hypoxia means there is circulatory failure due to which oxygen cannot reach the lungs because of cardiac failure. Then histotoxic hypoxia, and this is at the tissue level. And this histotoxic hypoxia can further be subdivided depending upon the site of interference. That means either it is extracellular histotoxic hypoxia, or pericellular histotoxic hypoxia, or substrate histotoxic hypoxia, and metabolic histotoxic hypoxia. The extracellular histotoxic hypoxia, in this condition, the tissue enzyme system of the body is poisoned so that the oxygen cannot enter into the cells. Like cyanide poisoning, in which the cytochrome oxidase enzyme system is poisoned. Then the pericellular, the oxygen cannot enter into the cells due to decreased permeability of the cells, like various anesthetics, halothane, carbon tetrachloride. It acts at pericellular level and they interfere with the entry of oxygen into the cell. Then substrate histotoxic, insufficient blood food metabolites. Their defective availability of the metabolites. Like we know that in metabolic processes, the glucose and the oxygen is required. So besides oxygen, the hypoglycemia, in which there is a reduced glucose, which will cause substrate histotoxic hypoxia. Then the metabolic histotoxic hypoxia, it means the end products of the metabolism, they are accumulated, they are not removed. So further metabolism is prevented by the accumulation of the carbon dioxide or various other nitrates causing uremia. Now talking about the physiology of asphyxia. It has been studied by various scientists, but the most important work is by Schwann and Brusser on dogs who induce mechanical asphyxia by breathing carbon dioxide and various gas mixtures, and experimentally studied the physiology. They found out that there is great survival time in depending upon various gases, that survival time is variable, but the pattern of cardiopulmonary physiology and biochemical responses, they are similar in all anoxic deaths. And this is the various enzyme processes which are going on interfering with the process of respiration. Now, it has been seen that unconsciousness occurs within 1 minute, and it has got 3 phases of extremely short duration, which are altered physiology, and these are known as stages of asphyxia. And these stages are 3 pathological stages of asphyxia, named as inspiratory dyspnea, expiratory dyspnea, and convulsions. And number 3, the respiratory paralysis. In inspiratory dyspnea, there is predominantly efforts to breathe in. In this stage, the efforts to breathe in, they are predominant. And this stage is also called as stage of forced respiration, and it is due to stimulation of the respiratory center, and the clinical picture of dyspnea is seen. This stage is characterized by signs of physiological reaction due to decreasing oxygen saturation of the blood and stimulation of the respiratory center. The breathing becomes rapid, deep, labored, and extra muscles of breathing, they come into action. Blood pressure starts rising, the pulse become rapid, and the person become restless, anxious, feeling of heaviness in the head, and ringing sensation in the ears starts appearing. The face, hands, fingernails, they appear blue. Then the next stage they pass into expiratory dyspnea. And in this stage there is predominant efforts to breathe out because now there is accumulation of carbon dioxide. In first stage in which there was effort to breathe in, that was because of reduction in the oxygen, but this stage is because of accumulation of carbon dioxide and efforts to breathe out. And this is also predominantly known as stage of convulsions because convulsions are seen in this stage, and it is due to cerebral irritation. And clinical picture of cyanosis and hypertension is seen: loss of consciousness, constriction of the pupil, and convulsions. Then this is due to lack of oxygen, retention of carbon dioxide, and pooling of deoxygenated blood in the veins and the capillaries. The expiratory type of breathing starts in this stage, and the efforts are to breathe out. Breathing become more labored, clouding of the consciousness, and convulsions may start, and sphincters become relaxed, passage of stool, and incontinence of urine. The face and the hands, they are congested and cyanosed. There is exudation of fluid, sometimes it is blood stained from the mouth and the lungs. The tongue is protruded and may be bitten between the teeth. Eyeballs become prominent. The convulsion contraction of certain groups of muscles starts, which is known as cadaveric spasm, and these cadaveric spasm is seen in extreme emotional deaths in which a group of muscles come into contraction, and usually the muscles of the forearm, there is grasping, anything which is in nearby become grasped, and that is the cadaveric spasm. It is the last volitional activity before death. They do not pass into primary flaccidity but cadaveric spasm, like in drowning, in mountain fatalities, person falling from the mountain get hold of the shoot of the tree and it becomes clenched in the hands. Then the third stage is the respiratory paralysis. In this stage, there is paralysis of the respiratory center, complete loss of consciousness, and the respiration become infrequent, shallow, and gasping, and it is slowed down till death occurs. Blood pressure falls, the pulse become imperceptible, muscles become flaccid, and the reflexes are lost. Pupils are dilated, there is terminal vomiting, and there is incontinence of urine and feces. The heart may continue to beat for 10 to 15 minutes after the respiration have ceased. Recovery from this stage is rare because permanent damage to the brain has been occurred because of anoxia. Now, what are the biochemical changes of fatal asphyxia? Because this process of interference with respiration has led to altered biochemistry, and this is the cycle which is causing the altered biochemistry. There is a reduction in the level of oxygen in the circulating blood and increased in the level of carbon dioxide. So first is a reduction in the level of oxygen, second is increased in the level of carbon monoxide, and there is lowering of pH towards the acidic side. And number 4, the blood sugar level starts rising, and there is altered exchange of sodium and potassium ions across the cell membrane. And this atmosphere will be causing cell to die. So this atmosphere of the cell is conducive to its death, producing pathological changes of degenerative nature. Now, the pathology of asphyxia. So, all physiology, all biochemistry now will be leading to changed histopathology, which will be pathology of asphyxia. And this pathology will be elicitable when we'll talk further as signs of asphyxia. So, pathology of asphyxia will be that these are actually histopathological changes in the tissues associated with lowering of blood oxygen level. And the nerve cell, venules and capillaries, they are most susceptible because of lack of oxygen. The nerve cell dies immediately. Small blood vessels responds by dilatation, precipitation of stasis of the blood both in the venules and the capillaries. And it will be leading to first pathological change will be congestion and cyanosis. This capillovenous engorgement with the rise in the level of circulating blood carbon dioxide will produce congestion and cyanosis. You can see the brain and various viscera, they are congested and cyanosed. Then edema. This is because of accumulation of the fluid. Anoxia increases the permeability of the capillary walls, forcing fluid out of the tissue spaces. And you can see this edema of the feet. And this transudation of plasma into tissue spaces is called edema. Then petechial hemorrhages. The capillaries eventually rupture, causing blood leaks of small size out of these vessels. They are known as petechial hemorrhages. Then fluidity of the blood. Blood in asphyxial death remains in fluid form. Why? Because the capillary endothelium under stress because of anoxia secrete excessive fibrinolysin and which keeps the blood in fluid form. So fibrinolysin released from the anoxic endothelium is responsible for fluidity of the blood in asphyxial deaths. So two main causes of asphyxia we have summed up. The two main causes, they are either the respiratory obstruction or it is the circulatory arrest. So two main causes they appears. And the both types there is decreased in the oxygen concentration and increased in the carbon dioxide. At autopsy, the analysis of the blood will tell whether the circulatory arrest or the respiratory failure was the cause of death. And if the concentration of oxygen is more than 25 mm of mercury, then the circulatory failure is the cause. And if it is less than 25 mm of mercury, then the respiratory obstruction may be the probable cause. Now, the circulatory arrest precedes cessation of breathing and a vicious cycle is set in. And this is the vicious cycle. The reduced blood supply leading to tissue anoxia, to capillary edema, post-hemorrhagic changes, further reduction in the blood volume and further reduced blood flow leading to reduced volume. So this vicious cycle will going on. And then the post-hemorrhagic changes which will be seen as reduced volume of the blood, stasis, edema, congestion and petechial hemorrhages. So these all post-hemorrhagic changes will be causing further reduction in the volume of the blood, hence aggravating asphyxia. Now, what are signs of asphyxia? These were the pathological changes and these are elicitable as signs which we elicit at autopsy. What are signs of asphyxia? They result from the body tissues when they undergo parenchymatous parenchymatous degeneration. That is, parenchyma become degenerated. So they will be predominantly signs of asphyxia which are specific. Specific mean specific to the cause, that at which level they are interfered with, and then non-specific. Non-specific signs of asphyxia are seen in all asphyxial deaths. So, the specific signs which are present on the locus, which are specific to the type of interference. Whatever is the interference at that site will be specific signs. In non-specific, they are cyanosis, congestion, increased permeability leading to edema, petechial hemorrhages, and fluidity of blood. So these are non-specific asphyxial signs which are seen in all asphyxial deaths. These non-asphyxial signs, they are developed when obstruction to the breathing is maintained for about 30 seconds. And these non-specific findings are more marked if the agonal period is prolonged. And they are also more marked if the obstruction is incomplete. Examine these findings in a good light conditions. They will be easily elicitable. Now, the first sign, cyanosis. It is due to the diminished oxygen tension in the blood and increase in the reduced hemoglobin. And this becomes apparent when at least 5 grams of reduced hemoglobin is present and the blood appears purple or dark colored. Cyanosis can be seen in the skin and in those organs where the venous and the capillary blood is abundant, for example in the lungs, meninges, liver, spleen and kidneys. This diminished oxygen tension results in the capillary dilatation and stasis. And the venous return to the heart is further diminished, pulmonary flow is reduced in volume, resulting in further deficient oxygenation. So this is the cyanotic nails. You can see the nails, they are blue. This is another picture showing the cyanotic tips of the fingers. The cyanosis is the result of this vicious cycle. Asphyxia leading to capillary dilatation, further stasis, further reduced volume of blood returning to the heart and reduced pulmonary blood flow, further deficient oxygenation and asphyxia. So this vicious cycle is set in. The limitation of this cyanosis are due to postmortem oxidative process. There is diffusion of oxygen from the surrounding tissues, and after 24 hours the cyanosis may not be present. So in all asphyxial death we should try that immediately the body should be examined. There may be absence of cyanosis in asphyxial death due to cold weather and it appears pink, because in extreme cold weather, because of the reduced temperature, the metabolism is not going on. Oxygen is there, but it is not being utilized in the metabolic process. So it remains in the fluid form in the blood and the blood appears pink colored. So cyanosis will not be appearing in deaths due to cold weather. Then precipitation of cyanosis depend upon the amount of blood in the circulation. If the amount of blood in the circulation is less, then the cyanosis will not be appear. Then the increased permeability, capillary permeability, how it is increased? Due to anoxia, the cement substrate between the endothelial cell, it is damaged and the fluid, it transuddates into the tissue spaces. Because of anoxia, the cementum, the cement substance between the cells, it become porous and transudate occur into the tissue spaces. Excessive fluid will be found in the serous cavities, that is the pericardium, the pleural cavities. The pulmonary edema may develop rapidly. And the limitation of this finding is the blood may be redistributed due to gravity and the rigor mortis. and the dilatation of the heart and pulmonary edema is common in many other diseases, but they can be differentiated. And in many asphyxial death, this sign may be absent. Now the congestion. Due to anoxia and reduced return of blood to the heart, there is pooling and stasis of blood in the venules, in the vessels. They appear dilated and prominent, and this can be manifested in serous lining, conjunctiva, and all the abdominal viscera. Then the fluidity of the blood. How the blood remains in fluid form in asphyxial deaths. In asphyxial deaths, the blood remains in the fluid form, and this depends upon the rapidity of the death. And this occurs due to the release of fibrinolysin from the endothelial lining of the blood vessels because of anoxia. So the blood remains in fluid form in asphyxial death because of a release of fibrinolysin from the endothelial cells because of anoxia. Then pulmonary edema. Pulmonary edema is common finding in many modes of death, but in asphyxial death, pulmonary edema is more marked. Each lung should be weighed and assess the degree of pulmonary edema. Then the petechial hemorrhages. They are also known as tardive spots. These are small, about the pinpoint to pinhead size hemorrhages, and they are easily elicitable because of rupture of the small blood vessels due to anoxia. The increased permeability of the capillaries, rise in the intracapillary pressure will causing the petechial hemorrhages. These spots are usually round, dark colored, and well defined. They are generally found in those parts where the capillaries are least supported, that is on the serosal surfaces, on the face, conjunctiva, epiglottis, serous surfaces of the heart, lung, meninges, and thymus. And this is a picture showing the petechial hemorrhages. And this is another close view. You can see the petechial hemorrhages. This is another picture showing the petechial hemorrhages. And this is on the serosal surface of inside the mucous membrane of the mouth. And the subconjunctiva, they have coalesced to find the subconjunctival hemorrhages. And this is the serosal surface of the lung. You can see the fine pinpoint to pinhead size hemorrhages on the lung surface. And this is the heart showing the capillary small petechial hemorrhages. The petechial hemorrhages are more pronounced in those areas where the intracapillary pressure rises rapidly, that is above the level of neck, for example in strangulation. And this is a case of stampede. In chest stampede and traumatic asphyxia, you can see the shoulders and the face is deeply cyanosed. And this is another picture showing the traumatic asphyxia. The upper half of the chest and the face is deeply cyanosed. They are better seen in fair colored persons and rapidly visible in fresh bodies. They disappear when the putrefaction sets in. The limitation of this is that they are observed in death due to convulsions, acute heart failure, and bleeding disorder of blood dyscrasias, but they can be easily distinguished from the asphyxial deaths. However, in these conditions, their distribution is generalized in contrast to the death from the violent asphyxial death because they are above the level of constriction. Tardive spots does not exclude death from asphyxia. They are rarely present in drowning because in drowning they are not seen. Thank you very much, and I will continue in the next lecture about the asphyxia. Thank you. Take care. Allah Hafiz.